Good morning, everyone. Thank you for just waiting until these last days of the conference. And today I'm going to talk about some aspects related to the hallmarks of cancer and how it's linked to the creatine metabolism with some latest analysis that we have performed recently. First, some disclosures here. I have just also acted as scientific advisor for several brands and also collaborate with some also private companies just talking about dietary supplements as well, and obviously being part of the scientific advisory board of Creatine for Health. So here is content of what we will talk today. So first, just describe some minimal details after what Dr. Balliman has said, everything about that, and has worked too much on that as well. Then I will remark some of the highlights of cancer metabolism. And we will deep into what is the association between creatine intake and cancer, if there is. And with this part, maybe I will focus on what are those maybe genes, creatine metabolism related genes that can be changing or have maybe different expression after some cancers. So I will describe the bioinformatics workflow that we have in our group with some of the National Institute of Cancer in Colombia and some other countries. And then just to prioritize some manually curated, and we will analyze some creatine signature that is available in the molecular signatures database. We will close with some future directions because this field, as we will see, there is too much to understand about the biology of cancer and creatine. And finally, some take-home message of this talk. So first, to describe the creatine kinase and phosphocreatine system, we know that this is a very important pathway and could be a hub of what it's maybe a chemo-mechanical energy transduction, because beyond the fact that it's a buffer of the energy production and ATP resynthesis, there is clear also evidence showing all of the aspects related to mechanics where the creatine kinase system is involved. So Dr. Balliman explained all of this in much detail. I want just to highlight some of the facts relating the creatine kinase isozymes that we have. So there are some cytoplasmatic, as you know, the one is the isoform of the brain and the other for the muscle. And those are dimeric, you can see it here in this representation. And there is an octameric that has some also expression differences between, for example, muscle, where there is the MTCK2, and in some other tissues where we can find other isozymes. But these are octameric proteins that are very linked into what maybe is all of the processing cellular respiration. It is close to the voltage-dependent anion carrier and also the adenine nucleotide transporter. So it's very linked in this intermembranal space in the mitochondria. And that's why it is participating in all of the process on controlling cellular respiration between inside the mitochondria and outside. All of these green sparks that you see here are those places where the creatine kinase colocalized and where it has a very important aspect in energy resynthesis. Could be in DNA replication or in the common what we saw every day when we are training, the muscular contraction, or even with some trafficking, that this is something that's why I was highlighting the fact of the mechanosensing activity. Several of these movements that we have of vesicles in the proteins could be clathrin mediated or not. They depend also on the energy for the movement and to make a real what we see processing clinical research. For example, the translocation of GLUT4 or some other for the exchange of different ions across membranes and several other things. So the fact that all of this is happening just shows us that the complexity of the biology in humans and what we now try to understand in a inference-based and also Bayesian way is that all of these status that we find in the biology are dependent on how they stand or they maybe deal with the stress, with external stressors or internal stressors. In this current paradigm that we are now in science, this is called the allostasis interoception paradigm. It is based on the free energy principle that was proposed by Friston and some others. It is now deep in more in biology as well, as we found that the allostasis is not only the stability through change, as was maybe thought in the beginning, but it's also the capacity to return to stability, and this is why they call it like physiological resilience. So these aspects are very important for life. So as soon as we maybe deal with some stress, and stress understood as a perturbation of a system, not only as a psychological stress that we are normally maybe conceptualizing this stress as that. But the stress could be mechanical, could be metabolic, could be psychological, obviously. So after one stressor just arrives, we have to, our body and all our cells, depending on which level of organization you are watching, we need to deal with that. So that's why there is a wear-to-tear response. And this accumulation of the load that we have is called allostatic load. It's like the cost that the system has to pay in order to adapt. So it receives these kind of stressors, and it depending on the energy status or some other variables, so it will have a response. So you can see here a systemic point of view, or cellular, or even mitochondrial. It depends on this fractalic way that is presented. So in all of those components that we are referring, the creatine metabolism is linked. As we show with the green sparkles there, we have constructed a bioinformatics and a knowledge-based pathway, just trying to compile all the aspects where creatine is increased and what it's maybe in some of those dotted arrows that you see here are probable also maybe outcomes that are taking place. But even the creatine transporter regulation, the activation of several pathways that are related to what we will see later in cancer as the MAP kinases, and several other factors related to the exchange of information between the inner and the outside of the cell. So why I'm highlighting this, and it's because it is too linked to cancer at the end of the day. Just in the way that the cancer exists, it needs to have all of these processes taking place. Energy production, some other things related to metabolic reprogramming, and immunosensing about the stressors. So we will check some of these hallmarks that were proposed by Hanahan and Weinberg several years ago. And we start with one of the first things that we know is the resistance to cell death that has these cells. As we are part of this nature that it's necessary to constantly do changes, we need to have some like clocks or internal regulators that help us to make the cells like replicated once at a time and with some different time courses. So there are several pathways that control this. The caspase 8 or the apoptosome and some other complexes that exist. The rest in peace kinases and several other like the Becklin or the P53 are controlling this resisting cell death. So they are in cancer, there are some changes where these proteins are not working very well. So the cells survive longer and they have this activity that we will check more. To use energy to reproduce or to replicate and proliferate. So the regulation that exists in the cellular energetics, we know it as the Warburg effect, is something where the lactate accumulates in more concentrations, and there is a dependence on the extramitochondrial pathways. So maybe the system is no longer, even when there is availability of oxygen, it is not using it as normal. So this is called the Warburg effect. So this is linked not only for the intracellular aspects of the cell, but also in the outside. So macropinocytosis, for example, is, let's think that these cells are like trying to eat what they can just to replicate and continue just progressing. So one thing is phagocytosis, where they maybe take and encapsule one of the substances that are outside, or bacteria, or any other stuff that could be. But in this case, the micropinocytosis is referring to volumes of liquid that are outside the cell and metabolites that are surrounding the cell. So it uses these kind of vesicles, also clathrin mediated or not, to embed some of these external resources, again, as trying just to recompensate always. And this also evolves with inducing angiogenesis, where there are obvious factors that we know already, like the HIF1 or some others, the vascular endothelial growth factor, that can change during cancer. And it's because there is like a switch to be angiogenic in the cell. So it needs to have more blood vessels to receive, again, some of these molecules that will help with the maintenance of the cell and the replication. And that's why several of the tumor angiogenesis just refer maybe as an important aspect of what is the difference between benign tumors and some malignant tumors. For example, the benign tumors, maybe they are in this latent state or dormant, where they don't have adequate supply from blood, and it's because maybe they don't have this progression of the angiogenesis process. Another important and hallmark factor of this is the sustained proliferative signaling. So in here is the activation of several pathways that we call it normally as anabolic. That's a term that maybe has been a buzzword sometimes with some people, that they just confuse what is also important for the cell, and it's not only the anabolism but also the catabolism. We need both to have this energy compensation according to the second law of the thermodynamics. So these pathways obviously need to help to create more proteins, or to differentiate, or to have intracellular or extracellular communication. I just want to also highlight that, remember that the mTOR, the mechanistic target of rapamycin, we normally find that people in exercise sciences or maybe in nutrition, they refer only always as mTOR, but there are complexes of mTOR. The one that we are referring to is the mTOR C1 or the mTOR complex 1, which is anabolic. The mTOR complex 2 is actually the contrary, is catabolic. And there are several other factors that are controlling their function, like the GATOR or the RAPTOR, and several other proteins that are related to energy, calcium, and some others. Enabling replicating mortality, it's one aspect also that it's happening in cancer. So cancer cells try to become like a stem cell, and with this phenotype, they can also perpetuate their existence. So they can differentiate to other factors, and in this case, the HIPO and the WNT pathways are the ones that are controlling all of these aspects. Again, this is so linked to energy metabolism. Actually, we can see here some of these proteins that we normally are talking about. So, and evading growth suppressors as well. So suppression is also controlled by the cell. There are some regulators of this that can block with autophagy, for example, or some others, that they can control that the cell just perpetuates. So these kind of systems are linked to the apoptosis, but are not the same. In this case, we are using like renewal, or certainly using materials that are already existing in the cell, like parts of the mitochondria, or some other membranes that are there just after some cleavage that exists. So in cancer, what happened is that it regulates the autophagy. So trying just to promote a microenvironment that is so linked to the immune system. And this is promoting growth and aggressiveness of those diseases. And talking about the immune system, actually there is an avoiding the immune destruction, where these cells maybe have some of these pathways that try to avoid the way that the immune system is controlling. As you already listened from Dr. Lily Yang, the CD8 plus T cells, those are controlling these kind of cells when they, maybe by surveillance, they are going around and when they check something, they try to solve the situation with those cells. But in cancer, there are some changes that what we call nowadays the tumors engineered or microenvironments immunology. This also has a different response in cancer cells. This is normally mediated by the STING pathways. It's like the stimulation of interferon genes. And these are some of these proteins that help us to promote this immune destruction of the cells, but they are altered during cancer. Finally, but not least, we have the genome affectation. So the genome is normally, as you know, also a very dynamic, complex, and important molecule that it spends too much of the energy. We normally talk too much about the exercise and movement across membranes and this stuff, but even the nucleotide phosphokinases that it sits in the nucleus and that are participating in the DNA replication, they spend a lot of energy and uses actually the creatine kinase system, and as I mentioned earlier. So there are changes not only for mutations, there are physical changes and the sequence of these nucleotides, but also some epigenetic changes. Maybe you have heard about methylation of the cytosines, sorry, and the other is the acetylation of histones. So those processes take place to modify certain genes, could, for example, silence a suppressor gene, or could activate one that it's promoting angiogenesis or these kind of things that I mentioned earlier. So at the end, we will have inflammatory status, where is pro-survival, pro-migration. It, at the end, results in metastasis. And using several of the things that we have maybe talked during these few minutes of the lecture, energy, oxygen, certain metabolites that exist there, and the renewal and recycling some membranes and some other factors. This at the end turns out in the metastasis process and invasion. So this also needs, again, energy and mechanics, because it needs communication between cells. There are several proteins in the extracellular matrix and integrins that help to do this. So at the end, all of these hallmarks that I already mentioned are metabolically linked to the creatine kinase and the phosphocreatine system. Because in each of these aspects, we have found also not only co-expression or co-localization, but also this importance of the energy and mechanics supply. And that's why maybe people has substantiate or base their claims that perhaps creatine, hypothetically, could be an enhancer or could be an accelerator of the cancer status. Nonetheless, there has been some studies that has shown the opposite. But we need to take care here, because we cannot say that it will not be linked or related, because as we already saw, it is. So there are several changes that we will see now with top 10 cancers that I selected also for aggressiveness and some impact and survival rates. And we will see how some of these proteins and genes are related. But in general terms, we have some data to say, for example, that creatine may prevent the body fat accumulation during chemotherapy in children. There has been some other data that you already saw, I guess that too much also from deep insights from Dr. Lily Yang regarding the anti-tumor T-cell immunity. And certainly one of the things that we look for is maintenance of the muscle mass in patients that are with cancer. This has been some reports about that, but also, as we will see now, there are some other trials that haven't found positive results or even no changes. Also, some claims regarding carcinogenic heterocyclic amines. Dr. Bruno Gualano and his group, several years ago, like 10 years ago, they published this paper as well. There were some sample of participants that they were supplementing with creatine monohydrate and even low or high dosages. that was not associated, or the detection of these heterocycline amides that didn't take place in association with creatine intake. So maybe that also it's a way to respond when we find this kind of claims that people saying that since creatine is an amino acid, non-proteinogenic molecule, so that it can increase these kind of factors. And other in vitro and preclinical research has shown these data that the Dr. Lilijan was showing. So there is some evidence about that, but we need more research just to continue having conclusive findings of this. Because, for example, recently, actually I see here Dr. Matt Stock, that is from the University of Central Florida. They did this short-term study. There was a 7 days dietary supplementation protocol with creatine, but they didn't find an influence of the creatine supplementation in some of the muscular performance variables that they analyzed. Too short, no? Maybe for the time, we can say. But also, sort of, some other trials has been published as well on this. For example, this was analyzing in a double-blind, randomized, placebo-controlled trial with using a loading phase and maintenance thereafter, and trying to analyze the loss of body mass in these cancer patients. They didn't find no effect of creatine when they compared to placebo, so they didn't maybe stop the weight loss that is associated with the cachexia that exists in these participants, even when they did a very long dietary supplementation. Even the survival median using Kaplan-Meier analysis, that those help us to see and evaluate the survival rates between two groups, they were almost the same for creatine and placebo groups, so there were no significant changes during this time. 500 days during that protocol, and no changes, I mean, in those factors that we were just describing, the loss of body mass and the anorexia and some other psychological components that they evaluate. So even when these have this data, recent and mostly this one, it's very interesting finding, because this is now not measuring what is happening when you are supplementing with monohydrate, creatine monohydrate, but in this case they are checking what happened when you analyze the dietary creatine intake, as those studies that Dr. Stojic has shown us during the conference, and they saw this reduction in the cancer risk for those who has a high dietary creatine diet. So they use with covariates or not the raw model, and what we can say is that at least for the NANES data, there is a secondary analysis that these researchers perform. Higher dietary creatine intake is associated with a reduction in cancer risk. So with all of this, we could say, obviously, what we are trying to promote, the safety of creatine, and also the importance to having less risk, in this case, for several diseases, not only for cancer, because there are several papers by Dr. Stojic that I recommend you to take a look, that find important aspects of health that are related to the good amounts of creatine in our diet. So this is an image just summarizing some of this information about the NANES studies, not only the growth in children and infancy, the taller in adolescents that we have found also with some of these data, the risks that are associated with issues in females, lower medical conditions, and improving the quality of life. So there are quite a bunch of papers about this. So you can just take a look later also. But then having this clear, that perhaps we can say that the creatine is a very important nutrient that we must have in our diets in the exactly amounts, because unfortunately several of the people we have maybe found in literature, they are not taking the the necessary amount from their diets. So we can say that it's safe, that it's not linked directly to the progression of cancer, and that in these kind of cohorts, there is certain potential to maintain muscle mass or avoid the accumulation of body fat. But still, there are further papers that are coming. Several protocols just were published the last year for creatine research and in cancer patients, in breast cancer, and some others, in survivals. So let's see what is happening with that, because actually we have also one project on that with the University of Deusto in Spain. And what we are also measuring is this long-term dietary supplementation and analyzing muscular variables and body composition and several other well-being factors. But let's just come back again to metabolism and check now what is happening with the creatine metabolism-related genes during different types of cancer. This workflow we have published in several types of cancer, in mesothelioma, in uveal, in also certain ovarian disorders. And what we are using normally is some prediction models for survival, where we the starting point could be different. In this case, we use manually curated prioritization. We will see it. But we can also have like an analysis of transcriptomics data to find differentially expressed genes. After this amount of bunch of genes that appears, we do a convergent analysis and then regression univariate Cox analysis that help us to make the prognostics. Another way is to also perform a meta-analysis of genome-wide expression studies that are available in public repositories, or taking directly signatures of certain genes that are available also in databases. The regular workflow that we follow is that once that we have the RNA-seq data, we extract those for the genes that are differentially expressed or that are a hallmark for this type of disease, a given one. Then we run the univariate Cox regression and do several models to divide them according to the risk, if high risk population or low risk. There is a very interesting paper that actually, as we will see, the GAMT gene is one of those that it's normally changing and differentially expressed in cancer, and that they are using elastic net regressions. Those combine the penalties of Rich and Lasso. Mathematically speaking, those are penalties for the softness of the model or the how hard we want to penalize each time that one of these factors are being linked or not to our independent variable. Then from this we extract the final model. It's an enrichment, and it allows an stratification at the end, because from this we can have the information, a low risk or a high risk phenotype. Some other secondary analysis like enrichment analysis with bioinformatics or ROC or some others, Kaplan-Meier, can be performed. And normally this is validated against a cohort that it's also an interesting way to test that this is not only using the full data available, but we can also have a comparison that it increases the robustness. Okay, but in this case, because it's not too much data available, we have one approach that is to use prioritization with certain genes, and I want to thank Dr. Balliman and Tarnopolsky and some others that has also recommend other genes, because we started initially with these four. But we now have tested some other that are obviously important for the creatine metabolism. I want to highlight this one. Maybe you see, if you are familiar, you will see here, this is the creatine transporter. But in this one, this is a protein transporter, a carrier that has been also linked recently and that it's already appearing in all databases to the creatine efflux in neurocognition. So there are some neurons that are expressing the oligodendrocytes that they express and release. This is a EB2 complete that missing part of the puzzle related to the synapses or this process of communication in the brain, releasing from vesicles creatine. So the missing part was like, okay, what is the protein that it's releasing from, not intaking, but releasing to other neurons? And it could be this one. Obviously, more data is still needed, but it was quite interesting that it's now being in curated databases appearing. And this is the set of cancers. We try to select some of those that are, as I was saying, more aggressive, less survival rates, or that has the worst impact in the person, in the cancer patient. So we have here glioblastoma, pancreas, lung, the colon, kidney, esophageal, liver, bladder, the stomach, and ovarian cancers. And we started running these models with the set of genes. Obviously, we maintain age because, as you know, and several researchers use this word like cancer is aging-related disease. And the senescence that it's occurring maybe is responsible of these disturbations that appears. And that's why it was maintained. In all models, it was obviously quite significant. And the thing is that here you have the set of the selected genes, and we will start checking some of these in greater detail. But we find, for example, here that the creatine kinase, the cytosolic, both subunits, the brain and muscle isoforms, and the creatine kinase mitochondrial 1b were, at least for the glioblastoma, significantly difference with the, or significantly associated, with the status of the disease. And with the survival that we will check in some minutes, the created with this data is that there are some secondary analysis that we perform that it's like this, but some bioinformaticians just take more data and from different cohorts, they validate the quality of the research, the place that it was published, and some of them, we already have some potential prognosis for different types of cancer. So those will be like my last slides. So I will show you that it corresponds also with our findings. In this case, for the lung adenocarcinoma as well, another isoform of the mitochondrial creatine kinase, and in this case, the creatine transporter, they were associated with this. And then again, we have the GAMT. This is an important molecule for the homeostasis of S-adenosylmethionine. So that's why it's maybe linked to the epigenetic processes that takes place in the cell. So changes in this one are expected and actually now associated with some higher risk of cancer in several samples. We found it here for the colon one. Again, here appears for the kidney. We have the isoform of the mitochondrial creatine kinase and the brain isoform. For the liver, we have this that was quite interesting, the fact that this transport, that is also a family from the transporter of carnitine, is also associated with the model. And we have, again, this molecule that produces, takes the guanidinoacetate acetate to take finally step of producing creatine. For the bladder, again, the same pattern. So we have GAMT and the mitochondrial 2 kinase. And for the stomach, again, we have the GMT with quite different, actually, with several samples. This is not showing here, but we use also several lines of in cancer that we have available also in the literature and extract where was these changes taking place in time course and also in differentiation with healthy individuals. And it's always the GMT like a hallmark. So we try to validate this, as I was saying, with the Human Protein Atlas, that it's a curated database that exists. So we check here, for example, for GAMT, and it's nowadays proposed as a prognostic marker for renal cancer. These are Kaplan-Meier plots. The way that we analyze this is favorable because the high expression is protecting or making the people more time alive. So the shorter or the way that this curve here is behaving is showing that lower expression of the GAMT, at least for the renal cancer, it's associated with death and less survival. With CKB, we have for colorectal cancer, renal cancer, in both of the cellular lines, as again a prognostic marker that it's being used and proposed. There are some other potential for the isoform 1A of the mitochondrial creatine kinase, especially for cervical cancer. And finally, this is a very aggressive, the glioma, and we have here the CKM2. So yes, definitely those findings enrich what Dr. Baliman just recommend to use other mitochondrial, obviously, isoforms of the creatine kinase. We see this very aggressive response. This is unfavorable. It means that higher expression of the CKM2, less survival rate, and again here in glioma as well. And just to complete the thing, just to say, okay, but it's this linked directly to creatine, so that's why we extracted the creatine signature. The creatine signature was taken from a database, the molecular signatures database that I mentioned at the beginning. So we have here a set of 28 biological elements, genes, all of them, linked to energy metabolism or the creatine kinase system. And then with this, we ran several analyses as well to find if the whole signature is associated or not with cancer, with the less survival rate in this case. So, and for colon, for example, we didn't find any association with that. And also for some others that I will just also complement these kind of things with the other cohorts that were validated too. We didn't find any significant association between the full signature of creatine, at least using the Kaplan-Meier plots. And actually, it's very interesting that these genes in recent years, for example, there was also a similar analysis like this. They call it pan-cancer analysis. They also extract tons of data from databases, especially focusing only in this one, in the creatine kinase mitochondrial 1A, and the link to certain tumors. They analyze also the full available data set that exists, that it's 32 types of cancer. And there is a very associated and as a prognostic marker that can be used, this gene. And the same with the creatine kinase mitochondrial 2. Also, it's linked to, in this case, colorectal cancer. In this case, they did a cohort of cancer patients. They just analyzed the expression of the data during a certain given time and make the Kaplan-Meier plots as well to check survival rates. So there is a thing there linked these creatine metabolism-related genes as prognostic markers of cancer. But again, I want to emphasize, it doesn't mean that creatine intake or creatine supplementation is changing or modifying these factors, because as far as we know, we have several trials that has used as well the creatine in different cohorts in healthy and non-healthy individuals, and no effects or association with cancer or tumorigenesis is taking place. So further clinical trials obviously are needed for this. And I want just as a future direction to mention this, because given the fact that is creatine kinase linked to the energy metabolism, so some researchers has started created with chemoproteomics some inhibitors of the creatine kinases. So they have created molecules that inhibit the creatine kinase, the cytoplasmatic and the mitochondrial versions, and they has been testing in vitro the way that it can suppress the cellular survival and promoting apoptosis at the end of the day. And as expected, hypothetically, this has turned also into a potent and selective target for the creatine kinases that avoid progression of those cellular survival and ends in apoptosis. But I guess that this also is still in their infancy. This was just published a couple of years ago, and they are just also contributing more, the laboratory of this researcher and some others. So at the end, we have these take-home messages. There are some clinical research showing that the creatine monohydrate is not associated with heterocyclid amines that are carcinogenic. The higher dietary creatine intake is associated with reduction in cancer risk. There is no direct evidence that suggests that creatine induces tumorigenesis. But we have these very important aspects to understand biology in cancer with creatine, where since it is a hub of the chemo-mechanical energy transduction, it should be obviously linked to the cancer hallmarks. And let's wait what happens in the near future with more data that will arise. So that's all from my side, and thank you so much.